Okay. You want to draw the
Let's take a nice random one.
Yeah.
This time we've got number 8.
Oh, 8. So what proteomics platform or technology do you offer services for? How do they work? Lori?
So right now, Psomagen offers Olink proteomics, and it works by a technology called proximity extension assay. And it's really quite clever. It was developed by in Sweden, and they will take antibodies that have 2 very close together epitopes that are on the same protein, and each antibody is tagged with a short DNA oligo. And when both antibodies bind that protein, the oligos get close enough that they can hybridize to each other. Then they can be extended and amplified, and you can basically quantitate how much of that protein is present by using either qPCR or sequencing. And there have really been some brilliant discoveries that have been made, particularly out of a huge data set at the UK Biobank. The NHS has socialized medicine, and they have a ton of patient samples and patient data. And they actually ran Olink's 3072 panel, which measures just over 2,900 proteins. And out of that massive data set, there have been a ton of really cool discoveries. And I encourage you to check out Olink Insight to actually get access to high-level data from that. The other thing that we do is we do support Infinity Biosciences, which basically uses either a whole peptide array or a whole protein array to screen humans or mouse samples to look for their immuno, basically looking to see what antibodies are expressed there. And it's also a sequencing readout. And then for the other, technology that we sort of support, our Macrogen headquarters, or previous headquarters, sort of sister company now, in Korea, offers Psomascan. Psomascan's adapter-based, and they say they have about an 11,000 plex. Illumina's currently partnered with them to validate all of their targets. And I think the current number after validation is about 6,000 plex. But this is pretty close to what Olink has in their portfolio, which is just over 5,400. But the cool thing for researchers is that there's no overlap, very little, which means to me that the aptamers are being able to bind to proteins that are resistant or not good candidates for antibodies. So it'll be a great sort of double tool set for people to have when that's through validation.
I see. That's great. So since I'm not directly in the proteomics space, what are some of the usage scenarios that are used often, addressed by or would be helped by these technologies?
I see their biggest utilization is really biomarker discovery. So being able to there's some incredibly cool papers that came out that I will absolutely go on and on about, so you have to stop me at some point. But being able to take a human plasma sample and basically screen that for potential biomarkers at, again, UK Biobank data, at a certain age, and then come back 10 years later and 20 years later and track that person and figure out. There was a study done where they are now able to identify people who are going to get Alzheimer's 10 years from the time that they get it. There's a protein signature just from plasma. And the best part about it is, so I don't know if you're familiar with antibody, Alzheimer's drug development, but there's almost every Alzheimer's drug fails. It gets to goes through phase 1, hits phase 2, and it just falls flat in its face. And I think the main reason for that is they're being developed against the symptom of the disease.
Yeah,
The
exactly.
brain
Yeah.
and the person is already really sick. They're not being developed against the cause. But now that there's a protein signature that can predict it before you're symptomatic, people can start targeting the cause of the actual disease. And that's amazing, because that gives you at least a better chance of being able to impede its progress or prevent it from even happening. So that's super exciting. Another group found, using the same UK subset of UK Biobank data, that they can predict dementia 7 years out, the most common kinds of dementia, like vascular dementia. And my favorite one that I can't wait till it becomes a test is this other team was able to identify, again, just from human plasma. And typically, I will also say that the vast majority of these Olink works in just about every biofluid imaginable, including tissue lysates. So, and I think Soma says pretty much the same thing. I'm not sure. I don't work with it, so I don't know as many details, but that's what I've heard. So again, human plasma sample, blood draw, minimally invasive.
Yeah.
There's a panel of 150 targets that can tell you, for men, whether you have stage 1 cancer. And when you add 10 more on, they can tell you where it is. And then there's a separate panel of 150 targets with no overlap at all for women that can tell you the same thing. Do you have stage 1 cancer, and where is it? And that is really the gold standard in cancer, quite frankly. It's early detection. But early detection without doing something that people are going to avoid, like a colonoscopy. No one says, oh God, I can't wait to get my colonoscopy, right? And you need a ride, anesthesia, and on. It's starting to be a blood test. I'm super excited about it because proteomics is doing what we were promised with genomics, right? When the human genome was sequenced, they were like, oh, we'll be able to predict everything. That didn't necessarily pan out, but and we got a lot of good information, a lot of cool stuff we've learned, and a lot of stuff that does, indicate or people's risk for disease and things like that. But at the protein level, it's amazing what you can do, what you can find out. It's super exciting time to be doing anything involved with proteomics.
Yeah, absolutely. So I have a question for you. So I feel like the Olink's approach of having basically a crutch man, two things together with a proximity measure, that sounds really innovative. The overall idea of just kind of proteomic total readout in blood, that sounds familiar, right? That's what Elizabeth Holmes went to jail for, right?
Yeah, that's the mass spec thing. If you want to do a total readout, there are mass spec assays that you can do, mass spectrometry, but it's really expensive. It's cost prohibitive.
Yeah, antibody-based approach is definitely much more scalable. So compare with what was Elizabeth Holmes' company name? Compare with that. They did just a sandwich, right? Theranos, right? So they were doing sandwich assays, right? Basically markers and sandwich multiplex.
I don't know what they were doing, but it wasn't. But the newest, so what I think is kind of like the newest, coolest technology that I've seen is from a company called Alamar Biosciences. Alamar. So what they're doing is they're starting with something that looks a lot like proximity extension. Okay, you have your 2 antibodies. They have a little tag on them, and they have to both bind to the one target protein, and they have to get close enough to each other so the oligos can anneal. But the similarity ends there. Instead of it being an extension, it's a ligation. So these two things are covalently bonded to each other now. And then the trick part that they've done is one of those oligos has a poly A tail and the other one has a biotin. And so now the next step is to wash. Because if you can't increase your sensitivity by
Oh,
making
that's
the
right.
binding tighter, you can increase your sensitivity by making the background lower. And so the first wash is to use oligo-DT to pull down that complex by the poly-A tail, wash, 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 anything that doesn't bind. And then you can just warm it up and it'll separate, because oligo-DT and poly-A just naturally want to bind to each other under certain temperature. And then the next step, now you have this complex that's been cleaned once, is to bind it to biotin to avidin, which is like a KD of like 10 or something. It's not going to come apart easily.
The strongest non-covalent reaction in the world.
Yeah, very good, cool. Now I learned something from you. So it'll bind, it'll wash, 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 and then detection is through sequencing. And they have attomolar sensitivity, and they have a 10 log dynamic range. Olink has a 7 log dynamic range, and I think, I should know, they have really good sensitivity. However, a lot of the neuroproteins that are circulating tend to be extremely low expressors, really low. And one of the things Alamar is really has done, they did great research and came out with a neuro panel that detects those. They're not going to get into the plex wars. They're being very smart, which I applaud them for. They're like, we're good at 200 or 300. That's high enough. But what we're going to focus on is making unique things that other people don't have. And also, their instrument was designed with clinical validation in mind, because all of the reagents are lyophilized. They come, you stick them into the instrument just like you're loading a sequencer, like everything's contained. The only thing you touch is the sample plate when you stick it in. That's it. Push play. So exactly what you want if you're going to do clinical. So I think it's a cool instrument. The Argo, of course, I don't know why they need that. And 2, I don't have $500,000. So I'm not going to be buying one anytime soon, but I am keeping an eyeball on them because it's a cool technology. And I think it would complement my Olink portfolio in a nice way.
Yeah, it sounded more like LabCorp Quest type of scenario. Is that a?
I think that's probably going to happen long term. Right now it's RUO. This is they're still building the business, right? They're still doing a lot of, when you first start out, you do a ton of custom work for people, like finding what's of interest, let them build their own custom panels and things like that. As you get a much bigger selection, that sort of scales down. They're still in that building up their repertoire. Though I feel guilty for not talking about mass spec more, but
the sensitivity of mass spec is kind of
exactly, and it's got its good points.
I'm in the antibody space, right? So I'm constantly have people who have some ideas talking to me about mass spec, how I should replace anything. It's like one of those things where you do a large screening, identifying some candidate, then you go validate those candidates with the more sensitive methods. It's kind of like that. It's definitely not a direct replacement.